Good morning, everyone. Let's go ahead and get started, finishing off our week. It always feels weird finishing a week on Wednesday, but here we are, Wednesday, February 3rd, 2021. We are going to finish up our discussion of blood vessels and depending on time, probably start a little bit on blood. I figure the more we can do today, the less we have to do on Monday, because remember, this is our last weekend to prepare for your first exam. We do still have a handful of assignments due on Monday. Your Unit 19 review is due. Your PhysioX Exercise 11 only Activity 4 is due, and your Hematology Labster is due. Which remember, for full credit, you must complete 100% and get 80% correct. Also, because we should finish early on Monday, there will be time for review for the first exam. Now, reviews are not me standing up here telling you what I think is important. That's what I do every day during lecture. Instead, reviews are a question and answer. Your opportunity to ask me questions about concepts you're not clear about, or especially for the first exam, procedures for the exam, or anything along those lines that will help you to be successful. And remember, if you don't ask questions at the review, then I assume you've all mastered the material and I make the exams harder. That leads us to 1 week from today, when you have your first lab and lecture exam. Again, there's no lecture on that day. We just have the lab and lecture exam. Remember, they're timed exams, so there's a set amount of time you must complete them within, but they also must be completed during class time. So again, if your lecture exam is 2.5 hours long to complete it, but you don't start it till noon, then you're only getting 35 minutes to complete it. I figure it's just like when we're in the classroom. If you came strolling into the classroom at noon and our class ended at noon 35, we'd have to kick you out to make room for the next class. So it's the same thing here. This must be completed during our class time. One thing that is different is you may take the exams in either order that you want. If you feel more confident about the lecture exam and you want to boost your confidence by taking that first, that you're welcome to do that. What I will say is that there aren't many problems with the exams, and definitely over the past year, Proctorio has gotten much, much better. But the few instances of problems that people do have, typically it does occur on the lab exams. So what I recommend is that you start with the lab exam first and start at the beginning of class at 8, so that if there are problems, you have time to work them out before the end of class. But again, there aren't a lot of problems, so do whatever you think is best. Then you get a whole week off. Monday the 15th is part of the President's Day weekend. And so to encourage you to be looking forward at the next material, because I would say mathematically, the next section typically has the lowest averages. It tends to be the least intuitive information, a ton of microscope stuff, a ton of cellular and chemical interactions. So it tends to be the more challenging section. So to encourage you to be thinking about and looking at this material ahead of time, I'm going to assign you the 2 pre-labs for the lymphatic system and our endocrine system. Those are pre-labs 21. Again, not the reviews. You'll be doing the reviews and turning them in, but this is the pre-lab. Remember, in our lab manual, there's 3 parts to it. There's the review at the end, which we've been doing and turning in. There's all the activities in the middle, but at the beginning of each unit is a pre-lab, typically labeling activities or vocabulary or things along those lines. So you're going to be doing the pre-lab for Unit 21 and doing the pre-lab for Unit 16, and those will be due at the beginning of the class on Wednesday, the 17th. Also, again, hopefully you know by this point in your 431 anatomy and physiology career, it is always important to be in class. However, it is especially important to be in class on Wednesday the 17th, because on Wednesday the 17th is when you'll be forming your groups, when you'll be selecting your topics for that 50-point group presentation that you're going to be doing. So while it is always important to be here, it's especially important to be here on that day, because if you're not here, you don't end up in a group. And if you don't end up in a group, you get a 0 for that 50-point presentation. So make sure you are here and available for that. All right. Questions on any of that? Is everybody asleep? I see no people on screen. I've heard nothing since I started talking. I'm assuming my microphone is on.
No questions.
All right, excellent. All right, excellent. Well, at least you're there. Perfect, excellent. All right. Then let's go ahead and dive into lecture. All right, we left off last time and we were talking about blood pressure and how to measure it and things along those lines. But obviously maintaining blood pressure, because we know it is that blood pressure that allows for the movement of our blood through our cardiovascular system, is vitally important. And so there are several factors that help us to maintain blood pressure. And the three main ones, as you can see here, are our cardiac output. Right, let's think about that. As cardiac output goes up, what happens to blood pressure?
Blood pressure decreases
as well.
Cardiac output goes up. Are we putting more blood or less blood into the blood vessels?
More blood.
More.
Pressure.
More blood. And if we're putting more blood into the blood vessels, what happens to the blood pressure?
It increases.
It increases. Excellent. It goes up. So if cardiac output increases, then blood pressure increases, and vice versa as well. If cardiac output goes down, blood pressure goes down. What about peripheral resistance? If our peripheral resistance, if the diameter of the blood vessels, the resistance of our blood vessels, if things like that increase, what happens to our blood pressure?
Increases.
Increases as well. And then, of course, if your blood volume increases, then what would happen to your blood pressure there as well?
Same.
Yeah, it would all increase. Notice all of these are basically directly correlated with each other. As one of these changes the others. Now, again, do we often see dynamic changes in blood volume?
No, not unless you get stabbed.
What were you going to say, Daniel? I thought you had it. You're right.
I said, not unless you get stabbed.
Okay, true. Oh, I wasn't thinking of it from that way. Yes, absolutely. Obviously, if you got stabbed and lost a lot or a bear with an axe takes a chunk out of your arm or something like that. I was actually thinking the other way. I was thinking of one of the things that many endurance athletes used to do is they would draw out a pint of their own blood a month or so before a race, giving themselves plenty of time to build back that blood. And then right before the race, they would pump that blood back into their body, what they call blood doping. The advantage of this is that it gives them more red blood cells, more oxygen carrying capacity, so they can get more oxygen to their muscles. So their muscles can produce ATP aerobically for a longer period of time without fatigue. Of course, taking a whole new pint of blood and sticking it into your arm dramatically increases your blood volume, which dramatically increases your blood pressure. And as we talked about, remember, high blood pressure can be very hard for the heart to have to work. And so it can cause a lot of problems and damage that way. But as long as you get that gold medal, of course, that's all that matters, right? So again, we have this direct relationship to these things, whether they change dynamically or not. Again, blood pressure is actually measured as the cardiac output times the peripheral resistance. And again, as we talked about, it varies directly. It's directly proportional to cardiac output, peripheral resistance, and blood volume. So these are the factors that influence it. Of course, we need to control these factors. And one of the ways we do that is with that neural control. Right? We have the two branches of the autonomic nervous system that control what again? Remind me again what are parasympathetic nervous system controls?
Mainly influences heart rate.
Right, absolutely. By directly innervating what structures of the heart?
Sinoatrial node.
Yep. And I'm going to cheat and abbreviate. And AV nodes. There you go. Excellent. So as you pointed out, this affects heart rate. And we know that heart rate is part of our, right? We know that cardiac output equals the stroke volume times the heart rate. So notice in this case, parasympathetic nervous system can indirectly affect blood pressure in that fashion. By controlling heart rate, it can influence blood pressure. However, when it comes to the sympathetic nervous system, right, what is it directly innervate? I did say that out loud, right?
The same things, including the Purkinje fibers.
Okay, excellent. So the SA node, the AV node, and the Purkinje fibers. Right. So as a result of that, again, it affects both heart rate and the strength of the contraction. As a result, again, this dramatically affects both heart rate and stroke volume. So obviously it dramatically affects cardiac output. And so indirectly, so let's say it this way, strongly, indirectly affects blood pressure. But what else does our sympathetic nervous system innervate?
Peripheral vascular resistance.
Exactly, absolutely. The smooth muscle of most blood vessels. And so if we can affect the diameter of the blood vessels, we are able to then directly affect blood pressure by changing the diameter of the blood vessels, which, remember, as we talked about, changes the resistance. And remember, as we talked about, even small changes in diameter can have a huge impact on the amount of resistance, and that resistance, as we talked about, affects blood pressure. Perfect. So again, I've written a lot here on the screen, but really there's no new information here. This is all stuff that we have already talked about, all stuff that we have already identified. So but it's still a good reminder for that. So, but I need to get it out of my way now. So let's go ahead and do this. Of course, we need to know how or when to influence it. And there's a couple ways we do this. There are chemoreceptors that can affect what is going to influence our blood pressure. But primarily it is controlled by baroreceptors. Baroreceptors are stretch receptors. Basically, these are sensory receptors that are constantly firing action potentials. I don't remember if we've talked about these before, so I will mention it again here. They're constantly fire action potentials. However, the rate of firing changes by their length. So what happens is if they're stretched out, the firing rate increases. And if they decrease in length, firing rate decreases. So they're constantly producing action potentials, but the rate of action potentials change based on how much they're firing. Now, when we talk about this relationship of blood pressure and blood flow, we have to think about this in terms of, let's talk about this baroreceptor reflex. Oops, I need that. I want that. So we have a blood vessel, and let's say that there is an increase in blood pressure inside of this blood vessel. So as a result of this, what's happening is that the blood is pushing more against the side of the blood vessel. And as a result of that, we have those sensors, those stretch receptors, those baroreceptors, stretch as a result of that. And when we stretch, what happens to their firing rate?
Increases.
Increases. Excellent. So again, if you think about it, we're talking about homeostasis here. We have a disturbance. I think back in terms of 430, we have our receptors that perceive that information. And where do the receptors send that information if we're thinking in terms of homeostasis?
Parasympathetic.
Well, you got the right. Well, you have the right idea. We're going to be sending information to the central nervous system. But if you remember back to 430, we talked about we need a command center, right? When we're thinking in terms of homeostasis. And you're right. These nerves are going to carry this information, as we talked about, to the medulla of the brain, where we process that information. Right, compare it to the information we know and make a decision based on that. And when we make a decision based on that, we then are going to send a signal to our effectors to influence and affect the change. So what would our two effectors be in this case?
Sympathetic and parasympathetic.
Well, those are the branches of the nervous system. You're right. We're going to use the branches of the nervous system to carry
The vagus nerve.
these signals. True, but remember, the effector is the thing that's actually going to influence the change.
Neurotransmitter.
So everything you guys are saying are absolutely correct. We are going to have nerves that are going to send the information. They're going to release neurotransmitters. But remember, the effector is actually the thing that brings the signal, that brings the body back into balance. Right? The vagus nerve, the neurotransmitters aren't the actual things that bring the body back into balance. So what are the structures that are going to be able to bring the body back into balance? What are we going to want to talk with? Smooth muscle, excellent. And more specifically, smooth muscle in the blood vessel. In the blood vessel. So the smooth muscle of the blood vessel is one of our effectors. And what are we going to want to do to that smooth muscle?
Relax it.
There you go, exactly. We are going to want to dilate it. When we dilate it, we increase the volume, that decreases the pressure. What's our second effector going to be?
The heart.
The heart, exactly. And what would we want to do to the heart in this case? If blood pressure is too high, what do we need to do to the heart?
Either decrease cardiac output.
Right, absolutely. Great idea. We want to decrease cardiac output. And we'll do that two ways. Reduce heart rate and reduce the strength of contraction. I'm all over the place. Excellent. So in that fashion, we then are able to bring it back in balance. Of course, if the opposite happened, right? If instead, whoops, no, I want that to stay there. If we had a decrease in blood pressure, what would happen to our firing rate then?
Decrease.
Decreased. Right. And what would we want to have happen? What would the effects of our effectors would we want to have occur as a result of that?
Vasoconstriction.
Yep. Constrict the blood vessel and
Increase the heart rate and contraction.
yeah, increase heart rate and strength of contraction. Excellent. Again, those should have been easy answers. It shouldn't have been somebody who was fighting for their life with a baby who had to answer those. But I appreciate that. All right, excellent. So Again, I've done an amazing job of drawing this on the board, but of course, we have the pretty pictures from a textbook that show this exact same type of process of how we're going to maintain that blood pressure using this pressure sensor. And these baroreceptors are not typically located just randomly throughout the body. There can be some in random blood vessels, but most are located, or there are a large number of them, let's say it that way, in the carotid bodies. Which, as we can see in this illustration, is where the common carotid branches into the internal and external carotid. And the other ones are located in the aortic bodies, which are located in the aortic arch. So those are two of the main areas where we have both baroreceptors and chemoreceptors that are giving us constant feedback of the condition of our blood.
Professor, could you go back one slide?
I can. There.
Where the drawing was.
There.
Thank you so much.
Yep. All right. Right. And then, of course, the other thing that influences blood pressure, and we skipped ahead a little bit talking about the baroreceptor reflex, is higher order processing. What do I mean by that?
Blood flows to more important systems.
True, that is true. We definitely want to direct blood to the areas that need it, but that isn't in this case what I mean by higher order processing.
Input from the central nervous system.
True, and not just input from the central nervous system, but typically we think of it as top-down stuff. Right? As we talked about, is you change the temperature of the body, heart rate increases and decreases and things along those lines. However, if you are watching a scary movie, can your heart rate change or blood pressure change as a result of that? If you're, it's 2 o'clock in the morning and you hear the vase outside crashing and the fumbling of the keys of the door as your loved one is trying to stumble in drunk on a Wednesday night or something along those lines, can that kind of thing influence your blood pressure as a result of that? Right? Absolutely. Fear, emotions, memories, all of these types of top down, right? Seeing that spider on the wall, all of these types of things are things that can influence it from basically your understanding of the world around you, your emotions, your memories, things along those lines. When we talk about higher order processing, those are typically the types of processes that we are talking about. All right. However, the nervous system is not the only thing that can control blood pressure. Obviously, we also have another system specialized for communication, and that is our endocrine system and hormones. Some of these are definitely ones we have already talked about in this class, or at least in 430, epinephrine and norepinephrine. And of course, what are the other fancy names for epinephrine and norepinephrine?
Adrenaline.
Adrenaline and noradrenaline. Yeah, noradrenaline, absolutely. Right? These are the ones that help us with our stress response, deal with stresses. So that increases our heart rate and the force of contraction. Notice epinephrine and norepinephrine cause both vasoconstriction and vasodilation. Is it random which is which? Do they alternate? On Mondays, they constrict the blood vessels, and on Tuesdays, they dilate. How do we determine what blood vessels are constricted and what ones are dilated?
Depending on the environment, what you're doing.
Okay, part of it is the environment. What else?
The receptors that they act on.
True, absolutely. The type of receptors they have, and the type of neurotransmitters the sympathetic nervous system releases. Well, actually, these are norepinephrine, epinephrine, so there are more hormones in the blood, although norepinephrine is also released as a neurotransmitter. But what else determines whether a blood vessel constricts or dilates? Let's think of it this way. What structures are going to see a dilation of blood vessels going to them?
The brain.
So that's a great question. That's a great question, a great answer for that, right? Again, think of it this way. That bear with an axe walks into the room. You have that stress response, right? You've processed the information, bear bad, axe bad, bear with axe, very, very bad. Is that usually the type of time where you're making deep philosophical discussions or thoughts or thinking about what you're going to do with your life or your long-term goals or anything along those lines?
No.
No, when that bear with an axe walks in the room, you basically have two choices. You can fight that bear with an axe or you can outrun somebody else in the room. Right? So typically when we're stressed, we don't see an increase in blood flow to the brain because we don't have to make deep cognitive processing when we're in that fear response. I always laugh at people who say, oh, I wait to write my papers at the very last minute because I write my best when I'm stressed. That's complete BS, right? Absolutely not true. We do not think well when we are stressed out. So, but as Allison pointed out, things that are going to help us to deal with that stress response, that fight or flight response, things like skeletal muscle, things like the heart, things like the lungs. And I can think of at least two more areas. What are the other two areas that we get a dilation of blood vessels going to them?
Eyes.
There is
they
a
dilate?
dilation of our eyes themselves. The irises dilate, so we don't necessarily have more blood going there. Do we get flushed when we get scared, or do the blood vessels in the skin constrict and we get pale as a result of being scared? Right. So here,
Constricts.
yeah, those. Well, let's think of it this way. Why do we go to the skeletal muscle?
So you can move fast.
Right, because we either have to fight that bear with an axe or we need to run. Either way, we're bringing all that blood to the skeletal muscle because what is it that skeletal muscle needs to be active? Oxygen, absolutely. And so that's why it's going to the lungs. But what else does it need? What am I using that oxygen for?
Glucose metabolization.
Yeah, making ATP. So the other thing I need is fuel. I need glucose. So something like the liver or something like our adipose tissue are places where we're going to mobilize our fuel, get our oxygen, so we can send it to the muscles to deal with that stress. Because that's what stress is all about. Stress biologically is to help us deal with those fight or flight, life and death situations, right? Because that's the way it used to be. 200 years ago, bears with axes roamed the countrysides. And so when we were scared, we literally were scared for our life. The problem is now the things that stress us out is that exam you have in a week, that 10-page paper you have due on Friday, right? Things along those lines. And if you have a 10-page paper due on Friday that's stressing you out, how do you deal with it? Well, you sit at a computer and you do this, right? At the computer. Unfortunately, the things that stress us today, we typically don't respond with in the same way with that physical activity. And that's the problem with this. I was glorious before I had kids, right? I had an 8-pack, I had hair, I had all of these things, truly, truly a glorious individual, right? But then you have those kids and you have all that stress that comes along with it, and you're releasing all of this energy and this fuel into your body to deal with that stress, and you're not doing anything with it. So what happens instead? You get that nice spare tire and the love handles and everything else that goes along with that, right? So typically what happens is things that are non-essential for that stress response are the things where it constricts: the skin, digestive system, kidney, things along those lines. Well, at least the drinking part. Yeah, fight or flight does turn into drinking. Things along those lines, absolutely. Right. So typically from a biological standpoint, that works too. The stress response, it helps us to basically mobilize our resources, mobilize our energy to deal with that stressful situation. So some things are constricted, some things are dilated based on that biological expectation of need. All right. Here is a hormonal process that I'm going to mention to us right here. We will talk about this in much more depth when we get to the urinary system. But our kidney, remember, our kidneys, as we talked about, when you are sitting here in class at rest, about 25% of your blood is going to your kidneys right now. So not surprisingly, the kidney has a tremendous number of chemoreceptors and osmoreceptors in it. And one of the things that it has also are pressure receptors. So when it sees a change in pressure, our kidneys are able to produce a hormone-like protein It technically is not a protein, and probably not a hormone, but it acts very much like a hormone called renin. That renin then leads to the activation of a hormone known as angiotensin. And angiotensin helps to activate a adrenal gland hormone called aldosterone. Like I said, when we get to the urinary system, we'll talk in much more depth about what these do and the full mechanism by which they work. But in general, what they do is they tell the kidney to hold on to more water. Our kidneys filter 200 liters of filtrate out of your blood during the course of the day. Do you produce 200 liters of urine during the course of the day? No, of course not. We'd all be sitting in the bathroom during this lecture if that was the case. Plus, none of us have 200 liters of extra fluid in our body that we can get rid of. So while we produce about 200 liters of filtrate, we only produce about 1 to 2 liters of urine. So over 99% of that is reabsorbed. And basically what angiotensin and aldosterone do is they tell the kidney, all right, bring back more water, bring more water back into the blood vessels. And if we bring more water into the blood vessels, what happens to the blood pressure?
Increases.
Yeah, more water back into the blood vessels. That leads to an increase in blood pressure, directly affecting blood pressure. Also, it tells the kidneys to bring back more sodium. And if we have more stuff coming back into the blood, what likes to follow stuff?
Water.
Water, exactly. So we bring back more stuff. The water follows it back in. And again, we end up with more water back in the blood vessels, and it brings our blood pressure back up. So this renin, angiotensin, aldosterone system plays an important role in reabsorbing water and sodium in the blood, which again, obviously affects blood pressure. Anti-diuretic hormone, released from the pituitary gland. Right? With a name like anti-diuretic hormone, guess what it does?
Water retention.
Yeah, it also targets the kidneys and tells the kidneys to bring back more water, which would increase blood pressure. Notice anti-diuretic hormone is also known as vasopressin because it also leads to a general vasoconstriction of our systemic blood vessels, which brings blood pressure up. Again, you may or may not have heard of anti-diuretic hormone before, but most of you are familiar with it. Anti-diuretic hormone plays an important role in our regulation of water, but one of the problems with our anti-diuretic hormone is it is affected by things like caffeine. So when you have that quad shot from Dutch Brothers, right, and you get all that caffeine in your system, you end up having to pee a lot for the rest of the day. Or when you go to the bar on Friday night and after your third Long Island iced tea, right? What happens when you go to the bathroom and break the seal? You're just peeing all night long, right? Absolutely, because alcohol and caffeine both suppress the production and the release of antidiuretic hormones. And don't even get me started on those four locos, right? When you get the alcohol and the caffeine together, you're just toast after that. Just might as well just live in the bathroom at that point, right? It's one of the things that leads to the hangover, the dehydration, because you're not holding onto water as a result of that. So this antidiuretic hormone plays an important role. And notice all of these, notice all of these increased blood pressure. Again, it is vitally important to maintain that appropriately high blood pressure so that we can keep that blood going. 4 Loco is a, it's basically like a combination of Everclear and Jolt Cola. Yeah, so if you're closer to my age, you probably get those references, right? It basically was a very heavily caffeinated alcoholic drink. Yeah, there you go. All right. So obviously notice it is vitally important to maintain high blood pressure. So if high blood pressure is good, then even higher is better, right? No, obviously we need to be able to bring it down as well. And that is where our friend, the atrial natriuretic peptide, comes in. This is a hormone actually produced by the atria of the heart. One of the interesting things about the endocrine system is the more and more we study it, there's still so much we don't understand about the endocrine system. And the more and more we study it, the more we find that almost every organ in the body has the ability to produce some hormones that can control and regulate things. And this is a great example. Remember, we have this huge pressure gradient. What was the pressure, the blood pressure of the blood when it leaves the left ventricle into the ascending aorta? What did we say a typical, normal, healthy blood pressure should be?
120/80.
Well, when it's leaving, when it's being
120,
forced out, 120. 120 is that, absolutely. And when it returns, when the blood has gone all the way through the system and returns to the right atrium, what did we say the blood pressure in the right atrium is? The blood in the right atrium is. Does anybody remember? There was that graph we saw. There we go. Rose got it. 0. When the blood gets back to the right atrium, the pressure in the right atrium, the blood should be 0. So what happens if blood pressure is too high? If the blood pressure is too high, then what happens is the pressure in the right atrium is greater than 0. And when that occurs, it causes the atria to stretch. And when the atria stretch, they produce this atrial natriuretic peptide. It sends this hormone, and this hormone targets the kidney and tells the kidney, release more water. Don't bring so much water back into the blood. Let some of that water, let some of that sodium go so the water will follow it. Let's get rid of that water so blood volume decreases and blood pressure decreases. So these types of hormones allow us to keep a very tight regulatory control on the blood pressure of our blood. All right, questions on that? All right, excellent. We have talked about ways that we can neurally and hormonally control blood pressure. Notice a lot of it has to do with regulating blood volume as well. And here's one of those fun flow charts from your textbook that kind of show all these things together. All right. Our goal, of course, is to maintain appropriate blood pressure, but there can be homeostatic imbalances when we're not able to. We can have hypertension, which is that high or low blood pressure?
Hi.
High.
It can be transient or persistent. What does that mean?
Comes and
Constant
goes.
high blood pressure or come and go.
Yeah, exactly. Absolutely. Excellent. Another important characteristic to focus on and emphasize when talking about and dealing with high blood pressure is whether it is a primary or what we call essential hypertension or what we call a secondary hypertension. Now, both are high blood pressure. So why does it matter whether it's primary or secondary? What does it even mean to be primary or secondary? Anyone know?
As a direct result rather than an indirect result.
I like that. That's an excellent way of describing it. I like that very much. Absolutely. Basically, with primary hypertension, the primary issue is high blood pressure. High blood pressure is the problem. However, with secondary hypertension, basically some other condition, you have some other disorder of the body, and that disorder causes the hypertension. Now, again, in both these cases, you have high blood pressure. The difference is how you're going to treat it, right? High blood pressure as a primary hypertension, there are many factors that can influence it. Diet, obesity, obviously things like age, race, and heredity aren't things that you can change. You can't change your genetics, at least not yet. But you could get into that less stressful job. You could stop smoking. You could lose weight. You could do all of those types of things, right? However, if you have an endocrine disorder, right, where you're producing too much aldosterone or something like that, that is causing, too much sodium to be pulled back into your body, doesn't matter how many times you say kumbaya, you're not going to be able to bring your blood pressure back down. So really, the primary difference, the main difference, let's say it that way, the main difference between primary and secondary hypertension is really how you're going to treat it, how you're going to try to resolve it. With primary hypertension, the problem is the high blood pressure. With secondary, you have some other condition that needs to be resolved. And when that is resolved, the blood pressure issue should go away as well.
Would
Now,
pregnancy,
yes, go ahead. I'm sorry, say that again.
would pregnancy be a secondary?
Yes. And in most cases, it goes away. Yeah, absolutely. So it's another great example. Absolutely. That would be definitely an example of a secondary hypertension. Yep. All righty. Excellent. Hypertension is obviously very important, but hypotension can be an issue as well. Again, it can be static, it can be persistent, or it can be transient. We talked about the example last class about orthostatic hypotension. This is where you get a temporary low blood pressure or dizziness from suddenly rising from a sitting or a reclined position. So as you change your position, because of the rate of your heart rate, again, your heart is all about producing efficient pumping of the blood. So when you're laying in a in a horizontal position, your heart isn't having to work as hard to pump the blood to your brain. But when you suddenly sit up, then it has to work harder to pump the blood up to your brain. And so there's a slight decrease in blood pressure, and you can get dizzy from that. However, hypotension can be a chronic issue as well. One example of a cause of chronic hypotension could be Addison's disease or poor nutrition. Those are things that can cause a persistent low blood pressure So things along those lines. And then, of course, we have things like acute hypotension. This can be an important sign of circulatory shock, where suddenly there's an instant drop in blood pressure. And again, this is what Daniel was talking about before with you get stabbed, or that bear with an axe takes a chunk out of your arm and a bunch of blood squirts out. However, those are the ones that aren't necessarily as big of an issue. Of course, getting stabbed and getting bitten by a bear definitely matters, but when people look at you, they know there's something wrong and they're aware of it and they can assess it. However, if you have someone recovering from surgery in the ICU and they suddenly have an acute hypotension, you may not see blood squirting out of them anywhere from the outside, but what do you need to be concerned about?
Blood loss inside.
Yeah, internal bleeding. You have to be worried about that internal bleeding. So acute hypertension is something that is monitored for very closely in someone post-operative, right? Because we want to make sure that everything's been closed up properly and there's no internal bleeding as a result of that. All right. Now, these types of deficiencies and others can lead to a failure of the cardiovascular system to meet the needs of the body. The tissues of the body need to have the appropriate amount of oxygen, need to have the appropriate amount of nutrients. And when they fail to reach that, we call that cardiovascular shock, right? Because we're not getting enough oxygen, we're not getting enough nutrients to the body. That becomes an issue because if you get less oxygen, as you know, we become less capable of producing ATP aerobically. And instead, we have to make it anaerobically. And when we make ATP anaerobically, we produce a toxic waste product as a result of that. And what was that toxic waste product produced by anaerobic respiration?
Lactic acid.
Lactic acid, absolutely. Lactic acid is damaging to the cells, can ultimately lead to cell damage and even death. And if that becomes extreme enough, we can get organ failure and so on and so forth. So we can have all these types of issues that come because of that. Now, again, this lack of ability to get blood to the tissues of the body can be caused by numerous things. One can be a hypovolemic shock. Hypovolemic shock is when there's a drop in blood loss. And again, there's the obvious things, the getting stabbed, the bear with an axe chewing off your arm, things along those lines. But the things that people don't think about sometimes is when they've had that gas station sushi, right? When you've had gas station sushi, you may have severe diarrhea for a couple of days, and that can definitely cause a dehydration. And that dehydration can lead to a drop in blood pressure, blood volume, which leads to a drop in blood pressure. Obviously, if the heart isn't working properly, that can cause issues as well, and that's cardiogenic shock from damage to the heart or or blockage of the heart or something along those lines. But it could also be called by the blood vessels. And we've talked about this a couple times. And this is why with my daughter, I carry an EpiPen everywhere I go with little, because as we've talked about before, I think I've mentioned in this class, I know I've mentioned the other one, my youngest daughter has a severe peanut and tree nut allergy. And so one of the things that can happen if she comes in contact with that special protein inside of a peanut, that she can undergo anaphylactic shock. With anaphylactic shock, basically two main things happen. One of them is that an increase in mucus production in the airways that makes it harder to breathe. That really doesn't have anything to do with the cardiovascular system. But the other thing that can happen is we can get a systemic dilation of blood vessels, right? So systemically, all the blood vessels dilate. And as we know, when those blood vessels dilate, resistance drops, blood pressure drops. And suddenly, not only can she not get air into her lungs, but now she can't get blood to her brain anymore. And those are two very bad things. So what do? You carry that EpiPen, give her that shot of adrenaline, right? So that it increases heart rate, increases the force of the contraction, constricts the blood vessels to help to bring blood pressure back up, and it dilates the smooth muscle of the airways, hopefully getting air into her lungs. Luckily, knock on wood, we have not had to give her an epi shot yet, but she's never had anaphylactic shock, even when she first we discovered it. But one of the frustrating things about these types of allergies is you could have 75 mild reactions to a peanut, and then that 76th time could be severe. So there's just no understanding it. So yeah, so that's an example. Anaphylactic shock is a great example of that. Lastly, things like obstructive shock is where blood can't get through a blood vessel because it's been blocked, like a pulmonary embolism or something along those lines. Damage to the inner lining of a blood vessel can cause clotting to occur. Those clots can break up and block a blood vessel. And if it blocks a blood vessel, then that stops the blood from flowing to those areas. All right. Excellent. Questions on any of that?
So we're only discussing types of shock that are like dealing with blood volume? Like we're not talking about septic
So
shock?
notice all of these, so these types of shocks occur because we are unable to get the amount of, yeah, there are other types, absolutely. There are other types as well, yes. But inappropriate firing of the vagus nerve, the vasovagal reflex is another great example of that, where someone gets an inappropriately high activation of their parasympathetic nervous system. And what ends up happening is their blood pressure drops because of that, and they pass out, from the sight of blood or something along those lines. So those types of things. So yes, there are plenty of other types of examples. These are just some of the examples of the types of shock that can occur in an individual. Absolutely. All right, so the last major concept we need to talk about for blood vessels, and from past history, the thing that for some reason seems to be most challenging for students is this idea of capillary exchange. Now, some of it may be because there's math involved. I'm not sure exactly what it is, but the two things that I will say is that for whatever reason, people have struggled with this in the past. So be aware of that. Make sure you understand these concepts. And they're almost certainly, I can almost guarantee with 100% certainty, you will have an essay question about this on the exam. That being said, I want to come at this information fresh. So let's go ahead and take our first break now. It's 8:52, so let's come back at 9:07. And at 9:07, we will pick up from here and talk about our capillary exchange. So any questions on that before we take our first break? All right, excellent. I will see you guys then in 15 minutes. All right, so like I said, for the cardiovascular system, the last major concept we need to talk about is this exchange that takes place in the capillary. One of the things we emphasize from the very, very beginning is that the exchange of all materials takes place at the capillaries. Notice here, you can see an interesting illustration from your textbook talking about some of the different ways that things can get in and out through vesicles, through simple diffusion. A lot of it goes through the intercellular cleft. And again, like most things we talk about in this class, it is all more complicated than we have the opportunity to go through. But in general, as we talked about, most materials are going to either pass through the intercellular class or through the plasma membrane just by simple diffusion. In both cases, whether it's sneaking through the intercellular clefts or through the cell itself, when it's diffusion, this of course is a passive process. And of course, what does it mean to be passive?
No does not use energy.
Yeah, it does not use ATP. There's no ATP used in it, which means it's primarily other pressures that cause the movement. Things like concentration gradients, or in the case of the capillaries, it's primarily the pressure changes between the capillary and its surrounding environment that cause those changes to occur. So let's talk about the pressures involved, because there's 4 pressures, 2 types of pressures. And I think for simplicity, let's just take a look. During the break, I spent a good 14 of the 15 minutes that we had of break trying to do my best job of drawing as accurate a capillary as I could. And here is the end result of that. So here you can clearly see this incredibly beautiful example of a systemic capillary. And of course, if this is a systemic capillary, then what is it that surrounds the systemic capillary? Exactly. It's like a Bernsey. Exactly. It's it's truly a work of art. What is that systemic capillary surround? What is it surrounded by? All right, quit admiring my art and tell me what the systemic capillary is surrounded by.
Interstitial space, the interstitial space, or the tissues that it is.
Both. Let's think of it that way. Absolutely. It is going to be surrounded by tissues, but you've got the right idea. In those tissues have interstitial fluid. So if you think about it, there is fluid that surrounds the capillary, that interstitial fluid. And then in our systemic capillary, of course, we have our blood. So we have fluid inside the capillary, we have fluid outside the capillary. So since we're dealing with fluids, the movement of the fluids and the stuff dissolved in the fluids are going to be controlled by two main types of pressure. Those two main types of pressure, which your book so eloquently describes as the suck and the push, are hydrostatic pressure and osmotic pressure. Hydrostatic pressure is basically the push. It is the force of the fluid pushing against its boundaries. Osmotic pressure is the suck, where it is the force that draws water to stuff. Right, back in your intro biology class, you heard about osmosis, how osmosis, water follows stuff. Where stuff goes, water follows. So we have these two main types of pressures, hydrostatic pressure and osmotic pressure. And since we have fluid in both of these environments, we are going to have these types of pressures in these environments. Here in the blood, nope, I don't want that there. Here in the blood, we are going to have a hydrostatic pressure. Hydrostatic pressure of the blood of the capillary, and we can hydrostatic pressure of the capillary for that. And essentially what this is the blood pressure. It is the force of the blood pushing against its boundaries. And because in this case, the boundary is the wall of the capillary and that boundary is leaky, this is pushing water and the stuff dissolved in the water out. So this is an outward pressure. Conversely, we know blood has stuff in it. And so in that blood, which of course, as we know, is a colloid. What is a colloid again?
Colloid is a solution with large particles in it that exert osmotic pressure on the surrounding area.
Yeah, basically what it means is it's water with stuff suspended in it. So there's a blood colloid osmotic pressure, where basically it is drawing, so we can go osmotic pressure, and again, we could do it of the capillary, so we'll do a C there. And this is the force of the stuff drawing water towards it. And so obviously in our capillary, this would be an inward force, drawing water into the capillary. All right. So far, so good. Stunned silence means yes, we understand everything entirely. Perfect. Now, is there water in the interstitial fluid?
Okay.
Yes.
Absolutely. So guess what? There is a hydrostatic pressure of the interstitial fluid. We can describe this as the hydrostatic pressure of the interstitial fluid. Right. And this is basically the force of the water pushing on the outside of the blood vessel. And since it's pushing on the outside of the blood vessel and the blood vessel is leaky, we would consider this an inward force trying to push water into the capillary. And is there stuff in the interstitial fluid?
Yes.
On occasion.
Absolutely. There is stuff there. We certainly wouldn't want it to be just deionized water. So of course there's going to be stuff there. So we are going to have an interstitial fluid, osmotic pressure. Right, we're going to have that interstitial fluid osmotic pressure. So osmotic pressure of the interstitial fluid, which is again the stuff drawing water to it. And since this is outside the capillary drawing water to it, this would be an outward force. So notice we have two inward forces and two outward forces. 4 different forces all influencing the movement of water and all of the stuff dissolved in that water into and out of the capillary. So if we want to understand how things move between the capillary and the tissue and the interstitial fluid that surrounds it, we need to do some math. The first thing we need to do is we need to calculate what our net hydrostatic pressure is. And of course, the net hydrostatic pressure would be the hydrostatic pressure of the capillary minus the hydrostatic pressure of the interstitial fluid, or the difference between those two. And because we care about what's happening with the capillary, we start with the capillary first. So it's the hydrostatic pressure of the capillary minus the hydrostatic pressure of the interstitial fluid. And that gives us our net hydrostatic pressure. Oops, and let's change the color of that to pink. And if we need to know the net hydrostatic pressure, then that probably means we need to know the net osmotic pressure. And again, we always care about the capillary first. So it would be the osmotic pressure of the blood colloid minus the osmotic pressure of the interstitial fluid. So now we have the net hydrostatic pressure and the net osmotic pressure. Of course, to truly understand which way things are going, we need to then put them together. So to determine, whoops, no, I wanted that color. The net filtration pressure, we obviously have to take the net hydrostatic pressure. From that, we subtract the net osmotic pressure, and that is going to give us our net filtration pressure. And with this net filtration pressure, as long as we set this all up properly, then you're going to get 2 results. You're either going to get a positive number or you are going to get a negative number. If you get a positive number, then this is going to be an outward force. Let's actually do it this way. An outward force. And if that is an outward force, then basically this location of the capillary is involved in filtration, things moving out. If we get a negative number, it's an inward force. And if it is an inward force, then we are dealing with reabsorption. So by throwing these numbers into an equation, we can then determine which direction materials, water, and the substances dissolved in that water are going to move. All right. Questions on any of that? All right. If we understand this, then all we have to do is figure out what the numbers would be, and we can plug and crunch. Now, I am going to give you typical numbers of a typical systemic capillary. But on an exam, for instance, could I give you any old random numbers I wanted for some capillary we find in one of these aliens when SpaceX finally gets to Mars and we're studying them? Could I then throw those alien numbers in there and you would still be able to figure out the direction that substances were moving into or out of their capillary?
Yeah.
Follow the same standards of life as on Earth.
Well, assuming they had blood and they had interstitial fluid, then absolutely it would work the exact same way, right? Or a special capillary in your body. Maybe a capillary in your spleen works different from a capillary in your liver, which works different from the capillary in your big toe or something along that, right? So again, know these equations, know how to plug and chug in them. I'm going to give you typical numbers, and those are important numbers to know, but it is also very important to know these relationships so that if I gave you any number on a not so theoretical essay question on the exam, you would be able to plug and chug and figure it out. All right. So
Excuse
let's,
me, Professor.
I'm sorry, go ahead.
Yeah, during the exam, will you be giving us like or providing us with any like formulas and stuff in case we have to solve for any equations, or we have to like
No,
just know?
any equations you have to have memorized.
Oh, okay.
So yeah, no, you have your, so you, no notes, no equations, no word bank, nothing like this. Again, this is an open brain test. Now, one of the things that I can encourage you to do is you do have that whiteboard. Yeah, I will do that. I will do that. But we also have the pretty picture from your textbook that shows it as well. We'll go back to that in a second. Is that you have that whiteboard that you can bring up. Or remember, the essay questions are going to have big spaces. So if you have all of your mnemonics and things like that, and you want to mind dump them onto the first question just to have them there, or onto the whiteboard so that you can have them there, you're welcome to do things along those lines. But no, that all the equations, all the numbers, all these types of things that you're responsible for, or information you're responsible for, you need to know this. All right. All right. Let's start hard first. Hydrostatic pressure of the capillary is the blood pressure. All right. Now, one of the things we know is this capillary, even though it's relatively short, if we want blood to pass through it, is the blood pressure going to be the same throughout the entire capillary?
No.
No, absolutely not. So at the arterial end, the blood pressure of a typical systemic capillary is, put this down here, 35 millimeters of mercury. Whereas at the venous end, the, I actually, I guess I should have put hydrostatic pressure of the capillary so we knew what we were talking about. I guess the color helps at least with that, is 17 millimeters of mercury. And if you remember, we saw that on that one graph. So when we talk about the hydrostatic pressure of the capillary, it changes from one end of the capillary to the other. Because again, that pressure gradient is what keeps blood going through it. Are we comfortable with that? Stunned silence means
To me.
yes. Excellent. All right, perfect. Now, what about the osmotic pressure? The blood pressure changes from one end to the other, but does the amount of stuff inside the blood change from one end to the other?
Maybe not from what, potentially. I mean,
Right.
it is the capillary, so it has to
You're
dump some of
absolutely
that stuff out.
Because it's passive, when water moves out, it takes stuff with it. When water comes in, it brings stuff with it. So overall, the relative amount of stuff in it stays the same. So regardless of which end of our capillary we are in, the osmotic pressure of the capillary is always equal to 26 millimeters of mercury. All right. Questions on that?
Always?
So again, is it always 35 for every single capillary, always 17 for every single capillary? No, these are average numbers, but the point is it doesn't change from the arterial end to the venous end. That's the real key to this. So throughout the capillary, it's going to be 26 millimeters of mercury. Okay, the only thing that changes from one end of the capillary to the other is the blood pressure, the hydrostatic pressure. Okay, excellent. All righty, perfect. We got that. Next, hydrostatic pressure of the interstitial fluid. This is the force of the water onto the outer wall of the capillary. Certainly, there is fluid in the interstitial fluid, but unlike the capillary where the water is trapped inside, is the water of the interstitial fluid trapped right up against the capillary where it has to bang its head on the wall of the capillary, or can it diffuse throughout the entire tissue?
The latter.
Yeah, it can diffuse throughout the entire tissue. So is it going to really provide any significant pressure against the wall of the capillary?
No.
No. So essentially, and again, it can vary from blood vessel to blood vessel, but essentially it's going to be 0 millimeters of mercury. It can go as high as, 3, 5, something like that, but typically it's 0. And so then lastly, we have our osmotic pressure of the interstitial fluid. Now, again, as we know, the water doesn't have to stay next to the capillary, but there is going to be a little bit of water next to the capillary. And of course, that water isn't just deionized water. It's going to have a little bit of stuff in it. This one can vary from actually a positive to a negative number. Well, actually, no, but in general, around 1. It's relatively small, millimeters of mercury. So these are good average typical numbers for this capillary exchange. And the good thing is now that we've done this, we can actually do our calculations. Now, notice what we see is that what is happening at the arterial end is different from what's happening at the venous end. So let's do the arterial end first. At the arterial end, we need to calculate our net hydrostatic pressure. And what would that be? I'll wait if you have.
35 millimeters of mercury.
All right, yeah, give me the equation though, because obviously on an essay question, you don't want to just give me an answer. You always want to show your work. So
35 minus 0.
35 minus 0, which of course equals 35. Excellent. What would my net osmotic pressure be? $25, but again, we want to show our work. 26 minus 1, which equals 25. And so what would our net filtration pressure be?
-1. Excuse me, 1.
1?
Yeah, because 1 minus 0.
No, we want our net filtration pressure.
Would be 10.
Yeah. Why is it 10? Again, show your work. Don't just give me answers on the exam. Show your work.
35 minus 25.
35 minus 25. Remember, it's your net hydrostatic pressure minus your net osmotic pressure. Notice I'm just using numbers here because I've already written out the equations here. On the exam, write out the equations and then plug and chug. And when you do that, you see that the end result is a positive 10 millimeters of mercury. So at the arterial end, is water and stuff leaving the capillary, or is water and stuff coming into the capillary?
Leaving.
Leaving. This is an outward force. And at the arterial end, filtration occurs. So at the arterial end, we have an outward movement of water and stuff. Does that make sense to everybody?
Yes.
Excellent. If that makes sense to everybody, then hopefully we can do the same thing at the venous end. So what's the net hydrostatic pressure?
17.
Again, give me the equation. 17 minus 0, which equals 17. What's our net osmotic pressure? There you go. 26 minus 1, which equals 25. And so what is our net filtration pressure? -17 minus, there you go, which is also exactly the reason why you show your work, Izzy, because if you had just put 17 minus 25 equals 8 on an essay question, then I will see that you understood the concept, you did everything correctly, and all you forgot to do is to put that minus, right? You're going to get the magic of partial credit. If you just have 8 written as an answer, I have no idea where you came with that number. Did you just forget the sign? Was it some other random way that you added or showed these things? One of the important reasons why it is important to show your work for full credit. Absolutely. -8. And if it's -8 millimeters of mercury, at the venous end, which way is water and stuff moving? Exactly. It is an inward force. And that inward force means that reabsorption is occurring. So notice we basically did this twice. There. We did the calculation at the arterial end. We did the calculation at the venous end. We were able to do this twice, and we were able to see different results. We have water and stuff entering the capillary. Right, so we actually got to do it twice. Yeah, it's not too difficult. You just have to plug and chug the information. So I could give you any 4 random numbers for hydrostatic pressures of the capillary, hydrostatic pressure of the interstitial fluid, osmotic pressure of the blood, osmotic pressure of the interstitial fluid, right? And you should be able to do the calculations. I could give you a number here of, right, 1,782. Right here, I could give you a pressure of 14. Here, I could give you a pressure of 375. Here, I could give you a pressure of 10,942. Oh, 42. And should you be able to plug and chug those numbers and tell me which way things are going?
Yes.
Yeah, absolutely. No, I obviously won't give you those kind of numbers, but you get the idea. All right.
Do we need to know these numbers that are on here though? As far
Yeah.
as like what is usually the pressure for the arterial end?
Great question. Yes, absolutely. You should know the typical pressures of a typical systemic capillary, but more importantly, you also need to know the equations for it as well. All right. Let me save this while I'm thinking about it, and let's do this again. Again, two types of pressure, hydrostatic and osmotic. Or again, as your book says, the push and the suck. Outward forces, as we know, cause filtration. And our two outward forces are the capillary hydrostatic pressure, which we know at the arterial end is 35 and at the venous end is 17. And the other outward force is the interstitial fluid colloid osmotic pressure, which, as we know, is essentially 1. Again, it can vary from 0.5 to about 5, but it tends to be on average around 1. We have two inward forces that cause reabsorption. Our blood colloid osmotic pressure, which, as we said, is 26, and our interstitial fluid hydrostatic pressure, which is essentially 0. But again, it can vary, can be positive, and in some cases, it can actually be negative. So we have outward forces and inward forces, and in a capillary, they differ at the two ends. So if we want to know what's happening in a particular location of a capillary, all we have to do is calculate the net hydrostatic pressure, calculate the net osmotic pressure, and put them together to produce our net filtration pressure, subtracting the hydrostatic pressure from the osmotic pressure. And remember, as we talked about, if you set this up properly, capillary first, hydrostatic first, then remember, as we talked about, a positive pressure is going to be outward, and a negative pressure is going to be inward. So as we talked about at the arterial end, we put all the math, and we get a positive 10, an outward force. At the venous end, we get a negative 8. An inward force. And if you don't like words, we have the pretty picture from your textbook that shows this as well. Questions on this?
So this is all to describe that phenomenon as blood is traveling down the artery or arterioles into the capillary bed, the kind of washout of water that happens as a result, and then the diffusion of products into the tissues.
Yeah, exactly. Remember, one of the keys that we talked about is the capillaries is where the exchange of materials takes place. So here at the arterial end is where things exit the capillary. This is where things like our oxygen, our nutrients, our hormones, right, etc., leave the capillary into the interstitial fluid. But as the blood continues down the capillary towards the venous end, then instead things get drawn up into the capillary. So these will be things like where the carbon dioxide, where the wastes, right? Where things, if this tissue happens to be an endocrine gland, this could be where the hormones go into the blood supply, etc. So again, it truly is an exchange of material. Things move out of the capillary and then things move into the capillary. So we are getting that exchange with the interstitial fluid. Basically, as we've talked about since the beginning of 430, we talked about the movement of water. What's happening in the plasma of the blood influences and affects the interstitial fluid of the body. And this is where we see that. This is where we truly see that it is indeed an exchange of materials that takes place. First things move out and then things move back in. However, there is one interesting point that we see about this. What is the outward pressure again?
The hydrostatic pressure.
Well, or the osmotic pressure. No, what is the net filtration pressure at the arterial end?
Positive 10.
Positive 10. What is the net filtration pressure at the venual end, the venous end?
-8.
-8. Notice they're not equal. So does the same amount of stuff get pushed out of the capillary as gets drawn into the capillary?
No.
No. Notice our capillaries are leaky. More fluid leaves the capillary than enters. Which is why, like the oil in your car, every 3 months you need to top off the fluid inside of your blood because you're constantly leaking like that radiator. You have to top it off, right? Is that what happens?
Yeah,
What
you have to
happens?
hydrate.
Say again?
Yeah, you have to keep hydrating yourself.
True, we do have to keep hydrating ourselves, but a lot of that is also what we're losing through perspiration, where we're losing through digestion, defecation, urination, sweating, things along those lines. But it's as a baby, you actually had 200 liters of blood. And now because it's been leaky, by the time you get to be adult, you're down to just 5 or 6 liters of blood, right? And by the time you become an old person, you have like 500 milliliters of blood in your body. All right, that's why grandma's so cold all the time, right? What happens to all this fluid that gets leaked out? Does it cause your hands to swell up like balloons? So again, you can't get that ring off when you get to the bar. We have more water coming out of our blood vessels than go back in. That's a leak. That could be a problem. How do we resolve this?
Is this where vasoconstriction potentially?
True, but you could only constrict it so much. As if every day you lost 100 milliliters of water by the end of the week, the end of the month. There we go. Allison's got it. If only we had an organ system that was responsible for collecting excess fluid in the tissues and taking it and depositing it back into the cardiovascular system. And lo and behold, that's exactly what our lymphatic system does. It's not super efficient. During the course of a day, your lymphatic system moves 2 to 3 liters of water. How much blood, how much fluid does your heart pump in a 24-hour period of time? All right, if it's pumping 5 liters a minute, what is it doing by the end of the day? Let's see, 5 times 60 times 24. What does that equal? A lot, exactly. A lot, absolutely. Your lymphatic system only moves 2 to 3 liters during a 24-hour period of time. That's not very much. But guess how much leaks out of your capillaries in a 24-hour period of time? About 2 to 3 liters. So the lymphatic system is sufficient, for the most part, for maintaining that consistent blood vessel volume, blood pressure, and taking that leaky fluid back. And the awesome thing about it is it doesn't just blindly just grab the fluid and put it back into the cardiovascular system. It filters it. It looks through it, looking for abnormal cells, looking for pathogens, right? Providing us with that immune response. However, the lymphatic system isn't perfect. If too much excess interstitial fluid occurs, what do we call that condition?
Edema.
Edema.
Exactly. We call that condition edema. Edema is when we get an abnormal increase in interstitial fluid. Injury can cause it. How does injury cause an increase in interstitial fluid? Anybody know? I know we haven't talked about our immune response yet, but I think most of you know.
Inflammation makes the capillaries more permeable, and so more fluid leaks out into the interstitial or extracellular space.
Absolutely. It dilates the blood vessels, bringing more blood to the area, and it makes the capillaries more leaky so that more fluid leaks out. And what actually is it that causes that dilation and that leakiness? What chemicals?
Histamines.
There you go. Histamines, exactly. Histamines are what cause that. So that histamine being released by the injured tissue leads to that increase in inflammation, leads to more interstitial fluid being in the space. However, now that we understand the capillary exchange, if you think about it, increased blood pressure, for instance, could cause excess filtration, right? Inadequate reabsorption, right? If there's a drop in osmolarity of your blood, that can cause inadequate reabsorption and things along those lines. All of these are things that could change the rate at which that capillary exchange is taking place. And then this doesn't even include things like lymphatic issues, problems with your lymphatic system or disruption of your lymphatic system. But those things can all cause edema to occur. And of course, when you have that edema, you get that swelling, often redness, pain, stiffness associated with it. However, in areas like your hand, your legs, your feet, things along those lines, some cases it won't be noticeable until it's about 30% above normal. Like I said, you may notice it's a little bit harder to get your ring off when you get to the bar, but it's not quite as noticeable in your movement of it. But as we also talked about, it also depends on the area, right? Do we want even 20% inflammation, edema occurring inside our cranial cavity? No. So remember, as we talked about, we have those huge sinuses to help to make sure that edema doesn't occur in those locations. All right. Questions on that? All right. So that is capillary exchange. And like I said, I guarantee you. Oh, great. Good question.
Doesn't it cause portal vein hypertension? And the result of that is increased fluid leaking into the abdominal cavity. And so as a result, you get like ascites. So the abdominal cavity just inflates.
Yeah, that seems reasonable to me. Yeah, well, absolutely. Now, again, obviously, the as we talked about, the swelling, the edema that occurs, for instance, in the feet, have more to do. Remember, we talked about how that increase in the size of the belly decreases the effectiveness of that respiratory pump to help to move the fluid back. And so, again, if you think about it, if you're having more difficulty draining blood from the veins, then that blood accumulates in the veins, it accumulates in the capillaries, blood pressure goes up in those areas, and more fluid is forced out. So absolutely. So yeah, so those are the things, those are the types of things that can cause excess filtration and lead to edema. Absolutely. All right, excellent questions. All right, excellent. And with that, we are done with the, oops, cool. We are done with our blood vessels. So the last concept we need to talk about, and we're just going to briefly start it today, is a discussion of the last major component of our cardiovascular system. And that last major component of our cardiovascular system is the blood. All right, we will start our introduction to it today. I don't want to get too much into it. We will finish a bit early today and still have not a full lecture on Monday as well. but let's go ahead, even though it's a teeny bit early, this is a good natural break point. So let's go ahead and take our last break here. There it is. So we'll take a 15-minute break, come back at 10:05, and at 10:05, we will restart our lecture. I will start the recording. All right, any questions on any of that? All right, grab one more stretch, go to the bathroom, grab something to eat. I will meet you back here in 15 minutes for our final stretch. So, last little bit we need to talk about for our cardiovascular system. We've talked about the heart, we've talked about the blood vessels. It is now time to talk about the blood. All right, we can't forget about the blood. Blood is a tissue. Anybody remember what type of tissue it is again?
It's a connective tissue.
Connective tissue. How do we define connective tissues?
They connect other tissues.
Okay, true. Yep, the building is tall because it is tall, right? How else did we define connective tissues? What are some of the characteristics we talked about that connective tissues have, right? If I think back to 430 when we talked about epithelial tissues, for instance, we know they're cell dense. We know they're on the surface, they're superficial. What were some of the key characteristics, and really the one key characteristic about connective tissues?
It has a matrix.
Excellent, absolutely. Like all tissues, it is comprised of cells, right? Because after all tissues are comprised of cells. But the other key thing with a connective tissue is that it tends to be more cell sparse, and there is that inorganic matrix that makes it up. Now, again, as we know, anatomists love to make rules and then love to break them. Is blood a true connective tissue? Does it have cells? Does it have matrix? Let's start easy. Does it have cells?
Yes.
Yeah. Like what?
Erythrocytes and leukocytes.
Excellent. Oh, I love it when we use fancy technical terms. Absolutely.
Thrombocytes.
Right. Excellent. All right, let's go ahead and do that. Erythrocytes, which of course is a fancy way of saying, and the appropriate way of saying what?
Red blood cells.
Yeah, they're the red blood cells, although you're absolutely correct. We want to use the correct anatomical terms always for things. So erythrocytes is 100% appropriate and accurate for that. Then I heard leukocytes, which again is the completely appropriate and accurate way of saying what commonly people refer to as white blood cells. Excellent. Thank you, Ash. And then I also heard thrombocytes, which is 100% the accurate way of saying what people commonly refer to as platelets. Excellent. And again, we always want to use appropriate terms. And in fact, red blood cells and white blood cells actually have cells in the name. So obviously they are cells. Or are they? Right? If we think back to 430 and we think back to what we thought of as a cell, right? Plasma membrane, nucleus, Golgi apparatus, rough endoplasmic reticulum. Do erythrocytes have all those things?
No.
No, they don't have a nucleus. They don't have most organelles. Basically, a erythrocyte is a big bag of hemoglobin. Now, at one time, it was a cell that had a nucleus and had organelles and had all of those things. But as a mature erythrocyte, it doesn't. So technically, it's not a true cell. It was at one point in its life, but as a mature cell floating around inside of your blood vessels, it isn't. What about leukocytes? Do they have nuclei? Can they divide? Can they metabolize? Do they have organelles?
Yes.
Yeah, absolutely, they do. Absolutely. What about thrombocytes?
No.
Yeah, thrombocytes are really cell pieces. They were ripped off. They were the arms and legs ripped off of a much larger, bigger cell. but basically they're small bags of cytoplasm held in a plasma membrane with a bunch of vesicles and other granular stuff in it. So again, they're arms and limbs, but they're not really cells. So notice 2 of the 3 things we're calling cells aren't true cells. Now, there are true cells in mature blood, and so that does meet the criteria, but we do have a minor vocabulary issue here. And so one of the things that our anatomists do to make things easier, harder, depending on how you think of it, is these 3 things collectively, rather than calling them cells where we have to qualify that they used to be part of or used to be or something along those lines, we typically use the term formed element. So these are all formed elements, right? And we use that term instead of cells, but basically formed element just means a cell or something that used to be a cell or related to a cell. And so all of these cell or used to be cell things are just collectively known as formed elements. But it absolutely meets the criteria for cells. So what about matrix? Do we have a matrix?
Is it the ground substance and the fibers?
True. So absolutely. Remember, matrix is made up of ground substance plus fibers. Absolutely. Excellent. Excellent. So let's make that part easy. Let's look at those. Does blood have a ground substance? What was ground substance primarily?
Mostly water.
Yeah, primarily it was water. Does blood have water? Yeah, absolutely. Excellent. So it's water and stuff dissolved in that water. So we meet the ground substance, but surely we don't have fibers in it. So it can't meet the criteria that way, do we?
Yeah, we do. They're dissolved as fibrin and they're only activated during clotting.
Excellent. Fibrinogen, you are absolutely correct, is the water-soluble form of a protein fiber known as fibrin. And you're right, fibrin plays an important role in our blood clotting process. And fibrinogen is the water-soluble version of it. So absolutely, while it may not seem like a tissue, it absolutely meets the criteria. It has a matrix. And of course, the name we give for this fancy water plus stuff plus fibers, what do we call the matrix of our blood?
Plasma.
Exactly. Plasma. So we have, let's be consistent. So we have plasma, we have formed elements, we have a connective tissue. Absolutely, it meets that criteria. And again, these are all things we talked about in 430, so there shouldn't be anything too new about that. But absolutely, it is a connective tissue, and in fact, it's sometimes referred to as vascular tissue, right? Blood is often sometimes referred to as vascular tissue because it is what is found inside the blood vessels. Excellent. As we hinted at, it regulates our extracellular fluid. As we just saw from the capillaries, basically what's in the capillaries leaves into the interstitial fluid. What's in the interstitial fluid leaves and enters into the blood plasma. So we regulate our extracellular fluid levels, all the fluid outside of our cells, by basically this exchange that takes place in the capillaries between our plasma and our interstitial fluid. And as we've talked about many times in this class, and here we in Sacramento know better than most, if you were to, for instance, drink a massive amount of water in a very short period of time to win a video game system, right, that can upset the osmolarity of our blood plasma and our interstitial fluid, leading to cell death, leading to organ failure, leading ultimately to death, because you didn't want a Wii to win a Wii, right? I know, but just like the jokes, I tell these things for me. If anybody else gets them, that's bonus. All of this stuff is for me. The references, the jokes, I do that to entertain myself. And if anybody else gets it, that's just bonus. Excellent. All righty. So, and then of course, the study of blood is the field of hematology. All right, excellent. So, as we've talked about, what is the function of our blood? Or really functions, plural.
Transportation of substances in the body.
Absolutely. Oops. Transportation is one of the huge ones. Now, let's think about this. With that transportation, let's think of two things. First, what is the medium for almost all transportation?
Plasma.
Plasma, absolutely. Plasma, almost everything that is transported by the plasma. And give me some examples of things that are transported. I think we did this earlier last week, but let's go ahead and do it again.
Albumin.
I'm sorry?
Albumin.
Okay, excellent. So proteins like albumin, absolutely, play an important role in maintaining the osmolarity, right? What else?
Chemical mediators.
All right, chemical signals, like hormones and other things like that. Excellent. What else?
Dissolved
Ions.
gases.
I'm sorry, dissolved what?
Dissolved gases.
Gases, excellent. Primarily which ones?
Oxygen, CO2.
Oxygen and carbon dioxide. Excellent. I saw ions. Excellent. Nutrients. All right. Wastes. What else? Heat. Excellent. I can think of one more good one. We just finished talking about it with not weeing for the we. Maybe it's too obvious. Water. There you go. Absolutely. Excellent. All of those things, other electrolytes, absolutely. And on and on. The list goes on and on and on. Absolutely. All of these are things that are transported. And as I mentioned, all of these are transported by the plasma, except one. What is the one thing on this list that isn't transported primarily by the plasma?
Gases.
Well, let's be more specific, because is both gases not primarily by the blood?
Yeah, oxygen and carbon dioxide, I believe.
Actually, just oxygen. Carbon dioxide, the majority is actually either dissolved in the blood or dissolved in the plasma, or it disassociates in the plasma, right? We can get into this here, right? Something mystical and magical happens when carbon dioxide is mixed with water. What's that mystical and magical thing that happens? Anybody know?
It dissolves or turns into carbonic acid, right?
There we go. Absolutely. Forms carbonic acid, which is H2CO3. And then, of course, like all acids, it disassociates into hydrogen ions plus bicarbonate. The vast majority of our, well, not the vast, but most of our carbon dioxide is transported, dissolved in our blood plasma this way as hydrogen ions and bicarbonate. Some of it actually dissolves directly into the plasma, and some of it gets carried with the oxygen. However, 98, 97, 98.5%, something crazy like that, of our oxygen is transported how?
Erythrocytes.
Yeah. And more specifically, bound to what inside of those erythrocytes?
Iron.
Hemoglobin.
And where's that iron? Yeah, excellent, perfect. Bound to the iron on the heme group of our hemoglobin. Excellent. Only about 1.5% of our oxygen is actually dissolved in the plasma. The vast, vast, vast majority of oxygen is actually bound to the heme of our hemoglobin in the erythrocytes, in our red blood cells. Excellent, excellent. So pretty much everything except oxygen is transported by our plasma. Now, again, that hemoglobin does also bind some carbon dioxide, but not the majority of it. And we'll talk about that a little bit later as well. All right. Any other functions of the blood?
Regulates body temperature.
Okay, excellent. Plays an excellent role in regulation. And not just regulation of body temperature, but as we also talked about, osmolarity of the interstitial fluid. Right, which of course affects the extracellular fluid, which affects the cells. So it plays an important role in maintaining homeostasis. So it helps us to regulate our body temperature, osmolarity, things along those lines. Obviously, again, this is primarily the function of the plasma that carries that heat. So we've talked about the function, there we go. We've talked about the function of the plasma. We've talked about the function of the red blood cells. We have the protection from pathogens. What primarily provides that?
Leukocytes.
Yeah. Our leukocytes provide that. Those white blood cells provide us with our immune response. We're not in the classroom, so I cannot get on my soapbox right now, but I can pretend to. You do not have an immune system, right? You have 11 organ systems. None of them is immune, right? Organ systems involve organs, right? You don't have immune organs. You have an immune response, cellular and chemical processes that take place that are associated with some organs, associated with the lymphatic system and things along those lines. No, but it would be really cool. I'll have to get one eventually. I had to get on it yesterday talking to my 4:30 class because we got on the coronavirus. I will save that lecture for you when we get to the immune response next one. Yeah, sure. Absolutely. The best dad jokes have props. Absolutely. In fact, how do you know when a joke becomes a dad joke?
When you're a dad.
When it becomes apparent.
Oh my.
Wow.
That was amazing.
You're welcome. Thank you. Thank you. All right. Excellent. So anything else as a function of the blood?
Clotting.
There you go. Absolutely. Basically, hemostasis, the ability to maintain our appropriate level of blood. And so we maintain that.
My beer is all fizzy now. Thanks, Austin. Oh, that's awkward. Please ignore.
That's all right. It happens to the best of us. Not a problem at all. I have teenagers, so they hate me, and I haven't seen them in 3 weeks. So otherwise, they'd be on my talking in front of me all the time anyway. So it's not a problem. All right, excellent. So hemostasis, you guys absolutely have the right idea. It helps us to maintain our blood volume, right, and not lose it when we're injured. So again, that blood clotting, that platelet plug formation, and blood clotting. Absolutely. So all of those are things that that are important functions of our blood. So we've written it on here. I can go ahead and erase that now, and we can see the pretty pictures. We did that. We did that. We talked about that. So absolutely excellent. Those are important functions of the blood, right? Of that connective tissue. Now, if you happen to have a bucket of it sitting right here next to you during the middle of class, how would you describe the characteristics of blood?
Viscous.
Excellent. So it has a nice viscosity to it. What else?
Sticky.
Yeah, stickiness, that viscosity, that stickiness, absolutely.
Warm.
Excellent, right? Typically, it's 2 to 3 degrees above our core body temperature. So when you cut yourself and it's pouring down your hand, you can feel that warmth from it. What else? Ash, you should save that for when we do the reproductive system. There's nothing better than having a big, huge hoo-ha on the board and then having a bunch of kids walk in. It's spectacular. So yeah, we'll save it for then. Then we'll have a bring your kid to school day. All right. What else? Warm, viscous.
It's red.
Okay, is it really red?
Looks red in the tube.
There you go. It looks red in the tube. Absolutely. But when we look at all those blood vessels, as we've talked about on every single chart, every single illustration, we know blood actually comes in two colors, right? Red and blue. Those we all know are the two colors of blood, correct?
Not exactly.
Not exactly. Absolutely. Not at all. Typically, when we use those red and blue, typically red is used to identify what?
Oxygen-rich blood.
True. Absolutely. Excellent. Oxygen-rich blood, whereas blue is our oxygen-poor blood. Absolutely. And the problem that you run into is often these are red is used for the arteries and blue is used for the veins. And where we run into problems is while that is true for all of our systemic blood vessels, it's not true for the pulmonary. So often our artists get in trouble when they work on the pulmonary. But again, this blue is just an illustration, a representation because of the way that light reflects through the skin. Veins tend to be more superficial, and when the light refracts out of them, it tends to have more of a bluish color. But you're absolutely right. The amount of oxygen that is bound to the hemoglobin changes the refraction of light. So when you have oxygen-rich blood, it is a very bright scarlet color. Well, it is tilted to the left, so I would say that it is a little bit on the left, so that's okay. Oxygen-rich blood is a much more bright red in color. And again, if you've watched those horror movies, right, that's what happens when the blood gets squirted out, or even when you cut yourself, it absorbs the oxygen from the air around you, and so it has a bright red color. However, and I think we talked about this in the last class, a lot of people have given blood, and when you are, well, true, if it's black and white, then that's different. If you're giving blood, when you give the blood, they're drawing that out of the vein into a vacuum-packed bag, and as you can see, when it is oxygen poor, it has a deep, dull red color. So definitely there does color to it. It is a reddish color that varies as you go from oxygen carrying content. Excellent. Anything else? Well, let's look at my list. Let's see what we came up with. Viscous, we got that. Sticky, opaque. Right. It has that metallic taste like pennies. Right, you run across some random blood on the street and lick it up and you'll taste that metallic taste to it. Slightly warmer than your core body temperature. A very restrictive pH range. Again, it is vitally important we maintain conditions of the interstitial fluid. We maintain conditions of the extracellular fluid around cells. So we have a very, very tight pH range to it. And then what did we say the typical volume was?
5 liters.
Yeah, about 5 liters depending on person, and we did the color. We did that already. Excellent. Because blood is so vitally important for maintaining homeostasis of the body. There are a tremendous number of negative feedback controls that help us to maintain both blood volume and the composition of the blood so that the body can function properly. There's, one of those crazy facts that every living cell in your body is something like 0.3 microns away from a capillary or something crazy like that, obviously we have this massive diffusion of materials and resources to maintain the homeostasis of all of the functional cells of the body. Now, if we were to take the blood, right, and again, here we look at blood makes up about 8% of all of the fluids and tissues of the body, about 8% of your body weight. And we were to stick it in a centrifuge and spin it around, one of the things that will happen is that it will separate by basically size. All of the formed elements get pushed to the bottom of the centrifuged tube, and all of the fluid and the stuff dissolved in the fluid, the blood plasma, is at the top. Notice blood is about 55% plasma and about 45% formed elements, although it can vary from person to person and other conditions, altitude, things like that can affect it, and we'll talk about that in a little bit. But notice actually when we spin it, it actually separates into 3 distinct regions. We have, as we mentioned, the upper portion, that is the plasma. But then notice the formed elements also separate. We have a thin layer known as the buffy coat that forms, and it is the one that is a very small, narrow region that contains the leukocytes and the thrombocytes. Whereas the vast majority of the formed elements, close to over 99% of the formed elements, are the erythrocytes.
1032.
So again, if we further divide it up, we can see the numbers. Notice of our plasma, it's a little over 90% water, proteins, and then a couple other things thrown in there for flavor. The vast majority of our formed elements are the erythrocytes, and a very small amount of platelets and white blood cells. And of course, we can further divide that stuff as well. Let's start first with the plasma. It's a little bit easier. Oh, and there's another pretty picture, sorry, showing the same thing. Again, about 92% water and about 7% plasma proteins. There are many plasma proteins. However, the most common plasma protein, as was mentioned earlier, is albumins. Now, albumins does a couple of things, but one of the important things to remember, as we talked about, is osmolarity of the blood is important. But what do I actually mean when I say osmolarity? We know osmolarity is the draw, osmosis is the draw of water to stuff. But when we talk about the osmolarity of the blood or the plasma or the interstitial fluid, what are we really talking about? What do we mean when we say osmolarity?
We're talking about its net hydrostatic pressure, or no?
Okay, let me ask you a question this way. I have 2 liters of water. Into one of these liters, I put 1 mole of glucose. And into the other, I put 1 mole of table salt. Do these 2 beakers have the same osmolarity or different osmolarity?
Different.
Why do you say different? They're both moles. They both have Avogadro's numbers worth of stuff, because that's really what osmolarity is about. It is the amount of stuff. Now, is it the glucose that's has the higher osmolarity because it's a bigger molecule?
Are we trying to, no, I'm not sure if we're differentiating between osmolarity or osmolality.
Well, it's the same for us. Which one has more, which one has the higher osmolarity?
I would say glucose does.
Well, but again, we're talking about the amount of stuff. So which one has more stuff in it? Do they have the
They
same
both
amount
have
of
1
stuff?
mole.
1 mole is 1 mole.
Okay. But 1 mole is 1 mole. But what else is happening? What happens to glucose when I put it in the water?
They want to have the equal amount on each side.
Well, that again, we're not worried about diffusion or anything along those lines. Glucose dissolves, absolutely. So basically you have, right, carbon ring surrounded by a shell of water, and it dissolves in there. What happens when you put table salt into water?
It splits into
Slowly
sodium and
dissolve.
chloride.
Yeah, it splits into sodium ions and chloride ions. So 1 mole of table salt is actually going to disassociate into 2 moles of stuff, right? If you don't like moles, let's talk 100. I put 100 glucose in water and 100 table salt in water. This is going to be 100 things dissolved in water, but this is going to be 100 sodiums and 100 chlorides, ions, dissolved in the water. And so this has more stuff. So really, it's not about the size, it's not about the mass, it's about the number of things, right? We're talking about the amount of stuff. And again, the amount of stuff inside, see, chemistry is made up. This is physics. This is not chemistry. This is physics. That driving force, all the stuff, anything in it. So the sugars, the glucose, the ATPs, all of those things are a total, the amount of stuff that is in the water, right? That makes it blood plasma. And one of the primary stuffs that plays an important role in maintaining the osmolarity of the blood are albumins. Albumins are a protein that play an important role in maintaining the appropriate amount of stuff in there. Another important blood plasma protein are globulins. Globulins are a class of proteins, and one of the more common types of globulins that we see are immunoglobulins. And while you may not be familiar with the term immunoglobulins, most of you have heard of them before, because what is the other name of an immunoglobulin?
Antibody, right?
Exactly. These are your antibodies, which play an important role in our immune response, our defenses of our body. We already talked about fibrinogen, that water-soluble protein that can come out of solution, plays an important role in blood clotting. And there are plenty more. And then what's left is about 1% of other stuff: electrolytes, nutrients, hormones, gases, yada, yada, yada. Your book's got a nice table that talks about some of the different components that make up our blood plasma, so we can see those things and the importance of that. Again, useful, important, not super exciting to talk about. What we want to spend more time talking about, where are we time-wise, is on the formed elements, as we talked about. Our erythrocytes, what we commonly think of as our red blood cells. Our leukocytes, there are 5 main types. Well, interesting question. What I would say is that I think having a low level of immunoglobulins in particular would be bad, because then you would have less of a time with your body's defenses, right? So again, higher immunoglobulins typically means that you are under attack, have an infection, but having low levels would make it harder for you to defend yourself that way. Remember, it's the fibrinogen that has to do with clotting, not the globulins. All right. There are 5 main types of leukocytes. We'll identify those a little bit later, but they fall into 2 categories, granulocytes and agranulocytes. And what do you think the difference between a granulocyte and an agranulocyte is?
One has granulations in it.
Yeah, exactly. Basically, when you look at the cell under the microscope, one of them has, I'll use stars because it's easier for me to do them quickly. One has a bunch of granules inside of them, and the other basically has no or smaller levels and smaller granules inside of it. And so we'll talk about those. There are 3 of our main types that are granulocytes and 2 that are agranulocytes. And then, of course, we have our thrombocytes as well. And here is the pretty picture that does a good job of showing us all of these main types. All right. Excellent. So what we are going to do, starting on, where are we at? Let me see where we're at. This is actually a good stopping point. So what we will do starting on Monday is we will finish, oh no, I closed it. Oh well. We will finish our discussion of blood by talking about all of the 5 main types of leukocytes, talking about the erythrocytes, talking about the thrombocytes and their functions, talking about hemostasis. Again, you have the lecture slides. What I will tell you is because it's right before the exam, we're not going to go tremendously in depth in the hemostasis processes. We will briefly mention them, and then we'll talk about the main concepts of them, but we're not going to go into the specificity, especially for blood clotting. As you'll see, it's this beautiful, very elaborate cascade effect. Your book actually does a nice job of describing the process. If you're interested in knowledge and knowledge is shake, then I think that that's something that's worthwhile looking at, but I won't hold you responsible for the process. We'll talk about the 2 main types and things along those lines. We will talk about that, but we're not going to go too much in depth in that. But again, you have the lecture slides that should guide you through the rest of this material as you're studying over this weekend. And then we will talk about that. The other thing that we're going to talk about, and again, from a histology standpoint, and that's the last thing that I wanted to kind of talk about for today. I don't actually think I have a blood slide. Let's cheat and use this one. So hold on, let me get that lecture slide back. I guarantee this is what I want. I love this picture. I like to believe that it is real, but I don't actually know. It's possible that someone has made it, but you know, they always talk about when you have, you know, a million monkeys punching on a million typewriters, eventually one of them randomly is going to hit the keys in the right order to have an Eminem song. And so it's quite possible that somebody may have found a view of blood that actually had all of our formed elements in one single field of view. So notice, obviously, we have, where's my, there it is, my spotter. These formed elements, by far the most common formed elements. What are these?
Erythrocytes.
Yeah, we got the erythrocytes. Again, they're avascular. They have that biconcave shape to them. Notice we have our 5 main types of leukocytes here and their different characteristics. Notice 1, 2, 3 have large distinct granules to them, whereas 2 do not have large distinct granules to them. Multi-lobed nuclei, different colorations to their granules, things along those lines, and we'll identify the 5 main types in the next lecture. And unfortunately, the only bad thing about this is there's this little piece of chewing gum or dirt or something that somebody got on the slide. Any idea? Is that what that is? Is that an artifact? Is that a little piece of dirt?
Platelet?
Yeah, it's a thrombocyte. Absolutely. That is actually our thrombocyte. It really is just a limb of a cell that has been ripped off and is in there. So indeed, we have all of our formed elements. So you absolutely, positively will be responsible for both knowing the function of all of these formed elements, but also being able to identify them using light microscopy as well.
That will be covered in the first lab and lecture exam.
Yes, because again, that's material we'll cover on Monday. So on Monday, we'll do this. I'm mentioning it here and pointing it out here, and then we'll talk about this more and identify them more and talk more about their functions. But absolutely, I guarantee on this exam, you will be responsible for microscopy of the blood and the function of all of these formed elements as well. Yes. All right. So we finished a little bit early today, but again, we've tried to front load things as much as possible. And unfortunately, like I said, normally we have more time to play with these things. You would actually be looking at blood under the microscope and doing differential blood cell counts. And like I said, we would have measured ECGs off of each other and things along those lines. So we try to mimic that as best we can with the Labsters and the PhysioXs and the things along those lines. But unfortunately, there's only so much we can do with this format. I don't know how much you guys as students are hearing about what's going on right now, but the union and the district are in discussions right now about plans for opening and when those are. The one piece of good or bad news, depending on how you think of it, is they have not made a decision yet on fall. Summer is going to be like this year has been, online completely. Including 430 and 431, but there is some discussion about a partial return, maybe for labs or even a full return in the fall. So they have been still in debating. At the beginning of the year, it seemed like everything we were hearing from the district is that fall was going to be online. Every indication was that. However, they continue to discuss these things, and my understanding is that no, we have not been, technically as an educator, I guess I'm in 1B, so I theoretically could, but again, Kaiser, who is who I have and most people are so strapped for vaccines, they're still in distributing just to 1A right now. So if they had it, I guess California, because California is at the 1B level, but it really it's location by location that way. But so no, me nor anybody that I know at the school has been vaccinated yet. So yes, we haven't been given that as an option yet, but it is still something that is being discussed. And so my understanding is the decision is going to be made by the end of this month. They purposely have delayed the release of the fall schedule. Summer, I think, is going to be released as it normally would be because it's going to be online, but they're delaying the release of the fall schedule, and they're still discussing the fall so that there's still a chance possibly we may be partly or fully online. So we'll see what is ultimately decided that way. I'm hopeful, but I'm also doubtful. If they've shown anything, they tend to be a bit conservative in the way they've been handling things from the beginning of this. So because it's been so slow to get the vaccines out. And again, remember the things about the vaccines, just because you may be vaccinated, all that vaccine does for you is reduce the severity of the illness for you. It doesn't stop you from making the virus. It doesn't stop you from spreading the virus to other people. And so then there's the whole issue of monitoring who can and who cannot get vaccinated. So there's a lot of complications in this issue. I'm thankful that they're putting time and effort into the discussion of it. But I think ultimately it's such a complicated issue. My guess is they're going to punt it for the fall. But we'll see exactly. It's all still very new. We're learning so much about it. And so that makes me think that they're going to punt on the fall. But the door is at least partially open right now. So that is good news. And we'll see what happens with that. All right. Excellent. All right. Any questions on that or anything else we've talked about? All right. We haven't left too much for us to do for Monday. So we'll cover that rest of the blood stuff, talking about these cells. Oh, and that's the other thing that I wanted to remind you as well. When identifying these histologically, obviously you need to know the specific type of cell. For the white blood cells, you need to know not only that they're generally all known as leukocytes, but you also need to know their specific names as well, specific types. So we need to know that as well for those. So the general type would be a leukocyte, and this would be an eosinophil. So I could ask that one of those two ways. As I mentioned, you need to know the function for these. But the other thing, and we're going to talk about this, is we need to also know the primary and secondary stem cells. for all of these as well. So again, that's something we'll be talking about in the next class, but know that on the exam, I could have a picture just like this with an arrow just like that. And I can basically, in this case, ask you 5 questions. All right, identify the general type of cell, identify the specific type of cell, identify the function of this cell, identify the primary stem cell of this cell, identify the secondary stem cell of this cell. All right. And then obviously for the thrombocytes and the erythrocytes, there's only 4 questions for those. And again, don't worry, we will go through all of this together on Monday, but I do encourage you, because this is your weekend to study before the exam, to at least use your lecture slides to move ahead from that. Oh, I was gonna say eggs. I think we spent a lot talking about ECGs. We interpreted the graphs. You had a homework assignment where you were interpreting the graphs. We talked about it in class. We talked about the blood flow. We did a lot of that. So are ECGs likely to be 1, possibly even 2 potential essay questions on the exam?
Like specific rhythms, identify specific rhythms, or just parts of sinus rhythm?
So I think it'd be more about interpreting it. Although, and the homework, did we talk about, I'm trying to remember what was on the assignment, I haven't looked at it yet. Was heart block on there? No, heart blocks weren't on there, but interpreting the different segments and intervals, I think is definitely something we talked about in lecture and was definitely a big part of the homework assignment and things along those lines. We didn't spend too much time, when we are in the classroom, we do, that was why I wasn't certain. When we're in the classroom, we spend more time talking about the physiological irregularities that can occur in that, but we haven't necessarily had the chance to do that in here in a meaningful way. So my guess is it'll be more about interpreting a typical ECG would be what I think would be appropriate that way. All right. Great question. Any others? And remember also, we'll still have time on Monday for a question and answer review as well. So that'd be the last thing that I'd encourage you to do over this weekend. Write down a list of the questions of the things you're not clear about or the things that you want more help with so that you can ask those questions on Monday so that we can answer those questions. This is not a unique situation to being online, but it's even more freeing when it's online. I have no idea what questions you are going to have on this exam. So I have no problem at all answering any questions you have, right? If you have a question about a process you want to go over together and that exact question ends up on your exam, awesome. Right? That's true for all the time because I want you to know this material. But in this case, it's completely liberating for me. I don't have to cage my answers because I'm afraid I'm going to give away one of the essay questions because I have no idea what's going to be on your exam. So ask me anything and I'll answer. All right. Questions on that? All right, so AMA Q&A on Monday after the rest of our lecture. All right, excellent. Guys, have a good weekend. Be safe, be smart, take care, and I will see you guys on Monday.